Welcome, everyone. I'm Manon Herzog from Polyplexus and will host today's episode of Talk Polymath. For those of you joining us for the first time, Polyplexus is a DARPA-funded platform dedicated to removing barriers and accelerating the research process. In other words, we drive the ideas that drive innovation. If you haven't already, you can join our community at polyplexus.com. So now, let me introduce our Talk Polymath guest. We are so pleased to have Dr. Farshid Guilak with us today. Farshid's resume is extensive and fascinating. He's both the Mildred B. Simon Research Professor of Orthopedic Surgery and co-director of the Washington University Center for Regenerative Medicine in St. Louis. He has also recently been nominated into the National Academy of Engineering. And closer to home, Farsh is a Polyplexus community member and contributor to our 2020 Evidence Madness. And with that, welcome, Farsh. Congratulations again on your recent nomination.
Thank you, Manon. I'm very excited to be here with you today.
So Farsh, let's start with your lab's primary focus on osteoarthritis. As I understand it, some projected 78 million Americans will suffer from it by 2040. What makes it so hard to find breakthrough solutions to this painful disease?
Well, that is a great question and something that I have been obsessed with for the past 30 years. The more we learn about this disease, it's always been historically dismissed as just wear and tear of your joints and unavoidable, and we're all going to get joint disease, either one form or another, osteoarthritis or rheumatoid arthritis. The more we learn about it, the more we realize that it really is a disease process that can hopefully be treated. It's not just an inevitable outcome of growing old. And what we've learned more recently, which is why it's so difficult to treat it, is that it's probably not one disease. It's a common endpoint where we reach this point where our joints are very painful. The cartilage, which lines the surfaces of the joints, has been degraded and degenerated and worn out. But there are many different ways to get there. And that's what we've started studying, are all the different forms of it. And because each form is a little bit different, the treatments are going to be very different. So we know that obesity is a major risk factor for arthritis. We know that joint injury, if you tear one of your ligaments or your meniscus, one of the stabilizing tissues in your knee, that's a huge risk factor. Or getting old is, of course, a risk factor for it. So the more we understand about the disease, the more we can learn how to interfere with it. The other very difficult part has been the cartilage that lines our surfaces is extremely hard to repair and regenerate. It's one of the few tissues in the body that has no blood vessels, no stem cells, no nerves, just no conduit to get anything there to heal it. So once you damage it even a little bit, it just gets worse and worse with time. So unlike a bone, which will heal itself, or muscle, which is full of blood vessels, very vascularized, and can repair itself, and a few other tissues like your liver that can repair itself, the cartilage has virtually no ability to repair. So all of these things together lead to a disease that's very slowly progressing, but very painful and very hard to treat.
Yeah, and hopefully by the time I am truly old, we have found a solution to this gnarly problem. But in the meantime, as I understand, in your current research studies focus on novel genome engineering methods with the goal of reprogramming stem cells. I must admit that sounds a little bit closer to sci-fi than medicine. Can you tell us more about it?
Oh, sure. Yeah, sometimes it does feel a little bit like sci-fi, but it's so exciting to work in this area because we're at this point of true revolutions in science. And you know, big revolutions are rare in science, as we know. We're generally, everything is fairly incremental. We build little bits upon each other. But some of the most recent technologies in biology and in engineering can come together now to allow us to do things like this. And these technologies have been For example, the creation of ways to make stem cells out of any cell, or what's called the induced pluripotent stem cell, which Yamanaka discovered in 2006, won the Nobel Prize. And then more recently, the ability to actually edit the DNA in a cell in a highly precise and reproducible manner. Now, editing DNA was possible before, but not in this manner where we can do it very rapidly, very efficiently. So something that would take years to do now, we can do in weeks using the method of CRISPR and CRISPR-associated proteins, which also won the Nobel Prize this past year. So combining these technologies have allowed us to create stem cells. And the beauty of it being a stem cell is then we can turn it to any other cell of the body. The definition of a stem cell is actually very simple. It's a cell that can divide indefinitely until a certain point where it can turn into a different type of cell, or differentiate, as we call it. So now imagine you can have an unlimited supply of cells that can grow up and turn into any other type of cell that you define, but now we can go in and reprogram their DNA. And the reprogramming can be very simple. We can give those cells the signals or cues to say, all right, you're no longer a stem cell, you're now a nerve cell, or you're a cartilage cell, or you're a bone cell. So we can grow huge amounts of living tissue, either outside the body or inside the body, by using these technologies. Now, the other major advance has been we can bring in engineering techniques now and not just change one gene, but we can put an entire gene circuit in there. So think of a really rudimentary computer that can sense something and respond to it, and we can now program that into the DNA of a cell. So instead of just passively responding to its environment, we can tell it to say, okay, if you sense some loading or some force, or you sense some inflammation, turn on this program. And if this stops, turn off the program. So really simple genetic circuits that we write as engineers, and then we can use CRISPR and other techniques to insert them into the DNA of the cell. Then we use one more engineering technique called tissue engineering, where we take those cells and we grow a piece of tissue like cartilage or bone or both together. And now we have the ability to restore tissues back in the body, but they have this whole other level of complexity where they can deliver drugs, they can sense their environment, they can fight inflammation, or they can grow more tissue when they need it. And that's where I think is some of the real excitement now. It just opens up all these possibilities where it is a bit sci-fi. It's your imagination is the limit now, because we can build cells that can do what you want them to. You can reprogram them. Of course, cells are extremely, extremely complex. And we're just talking about one fraction of a percent of the DNA that we can influence. But slowly, we can continue and build and make even more complex cells and more complex tissues using these methods.
It's amazing to think about how we might in the future really program our body in all kinds of ways. And is it fair to say that while you're exploring it specifically for this type of arthritis, the sky is really the limit, essentially?
Absolutely. We think of almost any disease where there's some problem either with tissue loss or with a feedback loop in your body. And you're talking now about the great majority of diseases and conditions that we have to deal with. Think of all the different situations where we have to restore some tissue or organ, or we have to correct a feedback loop. Like think of diabetes, where we have a feedback loop on insulin, or cancer, where cells are growing in an uncontrolled feedback loop, or just about anything. So we think, and we've started to apply these techniques to other diseases as well by working with collaborators here who are experts in those diseases. We can build the cells, we can build the genetic circuits. We call them smart cells, even though the smart is very, very simple smart. But just to be able to make one simple decision, and I often like to compare stem cells to teenagers. You want them to grow up, you don't want them to divide too fast, but you just want them to make one good decision. And if we can program our cells to make that one decision, to deliver the right drug or grow the right tissue, then we feel it's a success at this point.
Right. And we know bringing up children to even get them to making one right decision is a major challenge and is a matter of decades in some instances. So as you were discussing this approach, you already mentioned technology and such, and would like to hear a little bit more from you. I know your lab is very much focused on a multidisciplinary approach. Do you keep it pretty much in the realm of engineering, biology, and medicine, or do you actually go outside and maybe bring in collaborators we not typically associate with a lab like yours?
Yes, absolutely. We're excited in bringing anyone and any expertise that we can into this area, and I think that is my engineering background that pushes us to do that, which is a lot of our goal is problem solving, and this disease and related diseases are our problem. So rather than just focusing on what we know, we try to think, what do we really need to solve this problem? And fortunately, we're in an environment where we can go and talk to just about anybody here and say, oh, we need your expertise. Can you help us with this aspect of chemistry, for example? We need to make a chemical compound or a biomaterial compound that has these specific properties. Can you help us make it? Immunology, obviously, which is a branch of medicine and biology, but still very different expertise when we're dealing with immune cells in the body. We've actually started to work with bacterial systems and probiotics, for example, and understanding how the bacterial content of your body, the microbiome, as it's called, can actually influence arthritis, and how we can influence your microbiome with different diets, different components, and even applying these same gene editing methods to those bacteria that you can then put back in the body. So the multidisciplinary aspect continues to grow, and it's only bound by finding the right expertise and being able to convince them that your problem is important. And that has not been an issue. We just have so many fun collaborators here and great people to work with that let us go into these new areas and try really different approaches, again, because it is such a complex problem, and we have no cures or even real treatments beyond painkillers or diet and exercise for arthritis. There are no drugs that change the course of the disease, even now, not one.
Yeah. So definitely an area that still needs a lot of exploration and new ideas and hopefully new solutions. I know that you personally have certainly struck out way beyond your original field of engineering, and I would love to talk to you for a moment about the exhibition Design and the Elastic Mind. For those who don't know about the exhibition, it took place in 2008 at the Museum of Modern Art in New York. And I understand you collaborated with Paola Antonelli, the curator of the show, and would love for you to talk a little bit more why you accepted that opportunity and why you think it's important for scientists like you yourself to engage in this type of project.
Oh, that is a wonderful question and an amazing experience to be able to work with a true artist in this regard. And I've always, of course, loved art, and I was a member of the Museum of Modern Art for many years, especially when I lived in Manhattan and went to graduate school there. But even since then, we've stayed members, and modern art has been just a passion of mine. I have no artistic ability, although I love photography. But the combination of art and science has always fascinated me, and we're always looking for other ways to combine them for a variety of reasons. And I've been having these discussions with friends of mine, some of whom are artists, say, well, what is art and what is science, and what is the difference? And can you ever have bad art the way you can have bad science? We've established that, but it's very hard to think of situations where you would have bad art, as long as art does what you want it to, which is to either express yourself as an artist or to make you feel as the viewer of the art or the absorber of the art. And so when we bring science to art, it was very exciting to me because it goes beyond just those beautiful images that we take on the microscope, and has sort of become the mainstay of sci art, is who's got the best looking microscopy image. And those are very powerful, and I love those, and we still love making them in the laboratory. But it's also, how can we project science onto the realm of art to make us think of what's next? Both, science fiction that you mentioned is one aspect of it, but also, how does this scientific movement make you feel about the future and about what's happening now? So I think the way we ended up in this exhibit was, we had just published this paper on one of our first breakthroughs in recreating cartilage, which was to develop a method for three-dimensional weaving. So basically, we wanted to make a material to grow cells on that was tough, flexible, but really defined in an engineering manner. And we tried all different ways, like 3D printing and different biomaterials such as gels. And we figured out that if we could weave a fabric in 3 dimensions using hundreds and hundreds of tiny fibers that could go in the body and be compatible and safe, we could then put cells on it, grow the tissue, and then those fibers melt away and leave your replacement cartilage. So we had created this fabric out of hundreds and hundreds of these tiny fibers. And when we published it in a journal, in a scientific journal, the picture of the fabric made it onto the cover. And it was one of those beautiful, sci art images, electron microscopy. But I think it got the attention of the Museum of Modern Art and Paola Antonelli, because it brought together further concepts of what is, and in her mind, it was the elastic mind and elastic materials, and bringing sort of the mechanics of what we were doing to the mind, and saying that, okay, we can actually think of bioengineering and biomechanics as an art form as well as a scientific form. Now, we'd have to talk to her to really figure out what she was thinking and the thought process that went into it. But the exhibit overall just ended up being fascinating, and had a really broad display of all different areas of science and art, and the sort of physics basis to many of them of elasticity, but not just being beautiful images. They were very thought-provoking and very conceptually different from what scientists would normally see. And I was just really honored and delighted to be part of that.
Yeah, no, I feel exactly the same. And I think what you were saying, and what was so great about the exhibition as well, is that good design or art can actually really help in introducing very complex ideas to a broader audience and make it more accessible.
And also, yes, for science communication, and also provoking thoughts that you wouldn't normally have just from looking at a piece of science. Yep. That we as scientists often look at the data and we're constantly interpreting it in the critical manner of, what does this tell us? What question does it answer? What have we built if it's an engineering design project? And this pushed the concept of art beyond just something that's pretty to look at to something that is thought-provoking.
Yep.
Beyond the science. And I think that was what was so exciting to me about it, was walking through that exhibit and all of the other displays that were there was making us think about other things, not just science. And that's what art is for.
Yeah, agreed. So thought-provoking is a good segue into my next question. Sounds like you have really been collaborating with people from all kinds of fields. And can you tell us, have there been some particularly surprising or rewarding results that you didn't necessarily expect?
Oh, absolutely. And constantly. We're every day finding new things. Some of our best findings, or most interesting findings, I think, have been almost accidental, and have just been being at the right place at the right time, and then realizing it. And I think so much of science just happens, and sometimes we dismiss it as a mistake or overlook it because it wasn't what we were looking for. One of my best examples of that is that our laboratory was one of the first to really identify the fact that we have stem cells in our body fat. So just our subcutaneous fat that sits there, it turns out it has stem cells that can make more fat when you need it. So I had a friend of mine who had been working in the field of obesity, had started his own company where they were selling basically fat cells and the stem cells for fat to other drug companies for research. And we were having lunch, and he told me, yeah, we left a plate of these growing and we forgot about them for a few weeks. And when we went back, they had mineralized. What do you think that is? And it would have been very easy to just say, oh, something went wrong, throw them out. But instead, as we talked about it, we thought, what if they're actually stem cells? So they gave us a couple of flasks of them. We put them into culture, gave them a whole bunch of high-powered hormones and growth factors, and sure enough, they made cartilage in 2 weeks. And just a simple thing like that, a mistake that led to a little discovery that then led to a bigger discovery. That type of cell formed the basis of our research for about 10 years after that. Just being able to watch for those and be ready, always observing that when something's an outlier, it's not always a mistake, I think has been a really important lesson that I learned, and probably missed a few things along the way that would be great to have actually noticed.
Right. Yeah, I mean, who knew that fat could be that useful, right? Most of us
exactly.
are trying to lose it.
Because normally it's not. I mean, of course, you need fat to live, but it's usually not the best thing to have too much of in your body.
But the broader point being, we should really stop thinking about mistakes and more see it as an opportunity to explore further and say, well, we didn't expect this, but maybe it's actually going to lead us into a direction we would have never set out.
Exactly. Absolutely.
Yeah. So let's add to the mix. We've talked a lot about cross-disciplinary approaches, but as you know, there are a number of studies out there that suggest also multicultural perspectives are important, really across industries and of course in the sciences. Have you experienced it yourself that a multicultural and multidisciplinary team may have, been able to get to outcomes that you never expected?
Absolutely. I think that's such a great point. And, these days there's been such a nice emphasis on diversity. I think we've seen firsthand how diverse teams, whether they be multicultural or multigender, not having an all-male lab, obviously, but having people from different backgrounds, different countries, different thought processes, bringing those together always brings a difference in opinions and thought. And what I've always felt is that creativity comes from inconsistency in thought processes. If everybody thinks the same, you're going to end up at the same answer. And I even think individually, if you think the same way every time, your brain is so organized that you go down the exact same path every time, it's hard to be creative. That sometimes a little bit of chaos in your mind or in your interactions and cultural interactions and social interactions takes you to a slightly different path. And all you need is a little bit of nudge, like the butterfly effect. You just have one change in direction, one right at the fork instead of a left that you would always make if you were the same person. And bringing people together who have different backgrounds, I think, helps do that. As long as you're, obviously aware and respectful of the differences, which is critical. But with that, once you can communicate across different backgrounds, whether it's scientific backgrounds or cultural backgrounds or gender backgrounds or any other ones, I think it only helps to advance our ability to create and develop science.
Yeah, so I love this idea of a little bit of chaos, and we're coming up on time pretty quickly, but have a couple more questions that are maybe a little bit more future focused. And in terms of chaos, would love to hear from you who your ideal next partner might be. And the reason I say chaos is I know, for example, that Elon Musk, of course, is another member of the National Academy of Engineering. And I don't mean to say that is going to be your partner, but if you just had a magic wand, who would it be?
Oh, that is a great question. I don't know if I have a specific person in mind. I am actually kind of looking forward to potentially meeting Elon Musk, if nothing else, to see if I can convince him that these areas that we work on are potentially the future, and the fact that we can do things like create artificial gene circuits and decision-making capabilities and smart cells may be a way to go. I am very fortunate that I've had several collaborators that I've continued to work with for decades, 15, 20 years for some of them. And I think for me, the ideal collaborator is someone who does think a bit differently than I do, and not just another engineer. Although my closest collaborator has been my wife, Dr. Lori Sutton. We went to graduate school together, and we've worked together since then in all sorts of capacities, and we work great together. But I also find that if you work with someone who comes at a problem from a totally different aspect and has a different chaos in their minds and a different memory set and solves things differently, that makes the best partner, as long as you can communicate with each other. And usually I find these collaborations, the first step is to learn to speak each other's language. And if it's a biologist, one of my close collaborators was a neurologist, pain expert, Dr. Wolfgang Liedtke, and he always came at it from different angles than I did as an engineer, but we solved so many problems together. One of my best collaborators now is Dr. Christine Pham, who's a rheumatologist who treats arthritis patients. And every time we come up with a crazy idea like this, she reminds me, how do we get this to a patient in the next few years? And we have to retool and say, that's a great question. I'm not sure we can do it this way, but if we did it a different way, it might make it faster and easier. So I don't know if I've answered your question, because I don't know if I have an ideal person, but I think you look for opposites attract in this case, and people who will push your boundaries scientifically and intellectually and creativity wise, make you think about things that you wouldn't normally think about.
Yeah, people who disrupt your thinking. And with that, I also would like to explore with you, what do you think is next for biomedical engineering? And do you see any particular movements that suggest, there's a major breakthrough in X?
Yeah, that's a great question. And I think I'm biased. I think these areas that we're working in are really nascent. They're just getting off the ground because these technologies that we've developed have made things possible. The other area that's just hit our field by storm has been the concept of what we call single cell analysis or single cell sequencing. Normally, when we want to study a tissue or a group of cells, we'd have to grind them all up and then analyze that whole mixture, either as a chemical reaction or using imaging and microscopy. And now the techniques in the last few years have moved so rapidly that we can get thousands, tens of thousands of measurements on single cells, and that can be tens or millions of single cells at once. So the bottleneck there turns out to be the computational abilities. How do you handle now, you run an experiment and you get a terabyte of data off one experiment, which, when I did my PhD, I don't think I generated, an entire floppy disk of data by the time I was done. So you're talking about orders and orders of magnitude more data that are coming out, and being able to understand that and decipher it in a way that I don't know human minds can do by themselves. So we have to develop the software and machine learning algorithms to go through this level of data in a way that can tell us what's next. I think the combinations of those fields are just going to cause incredible breakthroughs in the next few years.
Yeah, I must admit, even in our conversation, we really just merely scratched the surface, right? I know there is so much more to talk about with you. We didn't get to the balloons behind you or Go's ear. I would probably suggest that people check you out. We have all your information on polyplexus.com. And, I cannot thank you enough for sharing some time with us. It's been absolutely fascinating, Farsh.
Thank you, Manon. My pleasure to be here with you.
Thank you. And to check out more cross-disciplinary conversations like the one we just had with Farsh, follow us on our YouTube channel. Thank you so much.